Hello everyone, welcome back. So this video is the second part of the Mendelian genetics. So we are basically discussing the chapter 5 of the NCRT, that is Principle of Inheritance and Variance. So Variation, sorry. So in yesterday's video, we have discussed basically what is genetics. We have discussed the term variation, and we have seen some terms like what are alleles, what are traits, and we also saw how Sir Gregor Mendel started his work and why did he think and how did he give a genetics point of view, and also how he worked on the pea plant or the Pisum sativum, and why did he choose the plant, and we saw the monohybrid cross in detail along with how to perform a back cross and a test cross. So in this video, I will be showing you in detail that how do you perform a dihybrid cross, and also I will be briefing you all up with the three Mendelian laws. So let's begin. I'm Janani Saptharishi and I'm associated with Unacademy. Unacademy is India's largest online learning platform. So, what do you get with Unacademy? You can avail daily live classes. Yes, every day you will be on top board with the educators and you will get access to all the daily live classes. You can clear your doubts, answer the polls, and you can uh, gain all the information there. And also with that, you will get access to all the live test quizzes, and we have structured courses with complete syllabus coverage. By that I mean, if you're, suppose like you're appearing for the NEET, so in one go you will get, not if you use my code, you will, it is not that you will only get access to the contents of biology and my lectures. No, you will get access to all the subjects you require to crack this examination, like physics, chemistry, biology, and all the tutors of physics, chemistry, and biology. There is unlimited access, that is, one subscription is enough to access everything, to all the live and the recorded courses, and you can watch it as at your comfort and ease. So you can prepare with the top educators, as I said, and it is your choice. With whomever you are comfortable at your ease, you can start preparing. So we have a very comprehensive syllabus, and at this time, at this situation, we all would recommend, sitting at home and trying to get maximum access to all the knowledge, information, and content. So all you have to do is go to the Unacademy Learning app and get it subscription. Once you get it subscribed, you can talk to us directly through the Telegram app. These details are given in the description box below. You only have to type the link and click on join. So these are the different fee structures what will be given as option to you when you start subscribing. So I recommend you the most about the 12-month course, because to prepare for an examination like NEET, you not only need 12 months in hand, but also it is the most economical. So it is just 15,000 rupees for 12 months. But if you're in class 12, if you're in class 11, I would highly recommend you the 12-month course. But if you're in class 12, you go with the 6-month or the 3-month crash course. And another interesting thing is, if you use my code, that is Janani, my name, Janani, if you use this code to get your subscription, you will avail an extra 10% off in all these courses. All right. Uh, that was a brief about Unacademy. Now let us start with the first two laws of the Mendelian genetics, that is about the law of dominance and law of segregation. So yesterday we discussed what is dominance. All right. So dominance is the term what we refer to as the traits or the characters which will express themselves in F1 generation. All right. And what it does it, this dominant trait will suppress the other. So what gets suppressed is the recessive trait. All right. I hope you can recollect this. If you have any doubts, you can type it in the chat box, and I will make sure that your doubts are cleared. Right? Hi, Ravi sir. Fine. So the first is the law of dominance. So law of dominance refers to the characters. That means the characters, by that, like of the pea plant, the characters are either it is tall, if it has a round pot shaped, if it has white or purple flowers, and it is controlled by discrete units called as factors. Factors are the ones which were later known as the genes. So these factors always occur in pair. All right. So what happens that in a dissimilar pair of factors pertaining to a character, one member dominates the other. So the dominant one is known as the dominant character, and the other one, which is suppressed, is the recessive. So, the law of dominance is used to explain the expression of only one, that is, the parental character in the monohybrid cross in the F1, and the expression of both in F2. So, it also explains the proportion of 3 is to 1. Yes. So what happens is that in the F1, only the dominant one is expressed. So in the X1, 100% the dominant gene is expressed. But that in the F2, so in F2, whenever we do a monohybrid cross, we always see that it is a ratio of 1 is to 3 or 3 is to 1. So here, both the traits are expressed, and we can see the proportion of it. Coming to the law of segregation, also called as the law of purity of gametes. What is a homozygous parent? So, a homozygous parent produces all gametes that are similar. So, a homozygous is that what will produce similar gametes, and the heterozygous is one that will produce different gametes, or two different which have some amount of change. Hello, Laksh Singh. What happens that Mendel states that in the law, according to his law of segregation, what happens is that the two alleles of a gene, when present together in a heterozygous state, the example of the heterozygous state is like tall and the dwarf, or the round and the wrinkled. All right, so these are the heterozygous conditions. They do not fuse or blend in any way. See, we saw what if I take the monohybrid cross. So let me just draw a small monohybrid cross here of the, that is the F2 generation of, let me say, the tall and the dwarf tree. So what happens? I'll either get, so I either get capital T and capital T, that is the complete pure line homozygous tall, or I get the capital T along with the small t, the heterozygous condition, or I'll get the complete recessive, that is the small t and the small t. So these are my four traits that I obtain. All right. So, do you remember that what we discussed yesterday is that all these four, out of that, These three, the first three, that is the complete tall, which has both the tall, capital T, and the capital T and the small t, these all are phenotypically very similar. That means all of them appear as tall. All these three appear as tall, whereas only the recessive trait appears as short. So this is the case. So what happens is that this is what is said, that they do not fuse or blend in any way. So there is no intermediate in between. So no in between character or a plant of a medium height is formed. So basically that is what is said in the law of segregation, that no fuse or blend, but they segregate themselves in a distinct way. That means टॉल टॉल जैसे एक्सप्रेस होता है एंड शॉर्ट शॉर्ट जैसे होता है एंड जब भी हेट्रोजाइगस बनता है तो डोमिनेंट वाला एक्सप्रेस होता है एंड रिसेसिव कंप्लीटली सप्रेस हो जाता है तो देयर इज नो मिक्सिंग अप ऑलराइट and this happens during the meiosis or the formation of the gametes. So each meiotic product or the gamete will carry only one of the trait. Hi Bharat. So moving forward, though the parents contain two alleles during the gamete formation, what happens is that the factors or the alleles of a pair segregate from each other and such that a gamete receives only one of the two factors. Yes. So when a parent has two alleles, what happens that during the gamete formation, these factors or allele, all right, of a pair will segregate from each other. That is what we saw like in the case of the tall and the dwarf one. Fine. So what happens that it is said that each gamete will receive only one of the two factors. Like is in the monohybrid cross of the height one, each gamete will receive only one of the two. That means it is either tall or the dwarf one. So what happens is a homozygous parent will produce all gametes that are similar. Obviously a homozygous parent is like, let's just take the example of the round one. So if it is R and R, all right, even when it will go undergo selfing, it will again produce the R1. All right, so since it is homozygous, it can only give or produce similar or the round gametes. A heterozygous parents will have two kinds of gametes, because heterozygous itself will mean that it contains different traits. All right. So what happens that these heterozygous gametes are produced in equal proportion, each having one allele. So when I say that, we'll look into it in detail. So what happens is that the segregation of these genes is a universal phenomenon in all the organisms reproducing by normal sexual method. So basically this segregation is ये सब हर एक ऑर्गेनिज्म में पाया जाएगा जिसमें भी सेक्सुअल मोड ऑफ रिप्रोडक्शन हो. ठीक है. So even in humans we see that we undergo uh, the segregation process. We can observe it in the chromosomes. All right. When the chromosomes split and they again segregate among themselves, they get reattached. So this is a very good example of segregation. See, in the first generation, it is a pure breed crossed with a pure, I mean that the dominant, completely dominant one crossed with a recessive one. All right. So the first generation, it produces four heterozygous plants or flowers. That is the pure, the heterozygous sweet red pea. So here all is red and the phenotypic ratio is completely 100% red. All right. Now, when I move to the second generation or the F2 generation, what I observe here is when these two heterozygous gametes are cross together, what we find is that we get a ratio of 1:2:1. This is the genotypic ratio. And the phenotypic is basically our 1:3 or 3:1. That means one white flower is observed. So here again we can see that how these gametes, that is the R, R, and again R and R. These four have segregated among themselves and they have produced uh, one completely dominant, that is capital R, and two heterozygous along with one recessive. So they have undergone the process of segregation. Now let us see what is a dihybrid cross. Hi Arun Patil. So now let us see what is the dihybrid cross and the law of independent assortment. So we have studied two laws up till now, that is the law of dominance and the law of segregation. Now once when we study the law of once we see that what is a dihybrid cross, we will come to know about the third law, that is the law of independent assortment. So, what is a dihybrid cross? It is basically a cross between two parents, all right, and what differ in two contrasting characters is called a dihybrid cross. So, whenever two parents will have two contrasting or two characters, they are called as the dihybrid cross. So, what happens is that Mendel conducted other experiments in which he followed the inheritance of two different characters. So, in one dihybrid cross experiment, Mendel studied the inheritance of the seed color, and in the other, he studied about the seed shape. So, these are the two different characters we are taking. So, two different characters refers to a dihybrid cross. So, the allele for the yellow color is the dominant one, and the green seed is the recessive one. All right, and the allele for the round, that is the dominant, is the capital R, and those of the wrinkled or the recessive is the small r. So Mendel has crossed these two. One is the round. You can see the shape of this pod. It is completely, around this, and in no way it has any wrinkles. So it is a round and it is yellow in color. So it's it's a true breeding plant. All right, that means from generations it has bred only with the round and the yellow. That means it has self-pollinated. And again, we have a true breeding wrinkled green seed. So this seed you can see is slightly wrinkled and it is green in color. And now we will cross these two. So in the F, this is the parental generation. All right. So whatever two we cross is referred to as the parental generation. And what happens in the F1? All right. So in the F1, only the dominant state will be observed. So F1, what we get is the round and the yellow. But just see that the F1 has also formed the heterozygous condition. All right. So basically, one gamete from round, yellow, and here from wrinkled and green has formed the F1 generation. So phenotypically, it is same as the dominant, or it will look completely similar to the round and yellow one. So now we will yes. So what happens is that the gametes RY, capital R and capital Y, along with the small one, will unite or undergo the process of fertilization. And this will produce the F1 generation. So, yes, now the F1 generation will undergo a process called as selfing. That means this F1 is again crossed with itself. Alright, again made to self pollinate. So now we will write all the gametes possible, see, as they undergo the law, under the law of segregation, we have to segregate it according to all the probabilities. So here we have capital R, capital Y, capital R, small y, the small r and capital Y, and small r and small y. So similarly, we write it in both the side, and what we get is this seed is completely round and yellow, and we have many round and yellow seeds. But see, only this one is a pure, or I can say that the completely dominant one, that is genotypically it has both the capital R and capital Y. So here, though all these are in the heterozygous conditions, though the dominant trait is expressed phenotypically, but genotypically it is not completely dominant, or I would say that it is a heterozygous condition. Similarly in these, all right, here, and this is again a completely recessive one, that is the small r and small y. So now when you start counting the phenotypic ratio, what you will observe is that the number of the yellow and the round seeds, that is 1, 2, 3, 4, 5, 6, 7, 8, and 9. So these are the yellow and the round seeds. Alright. Then moving to the yellow and the wrinkle. So we can see a 1, 2, and 3. So these are yellow and wrinkled. Alright. So this the yellow, sorry, wrinkled and yellow. Yes. And now what we come to is what is green and round. So here again we can see 1, 2, and 3. And what the recessive one is left is 3. So that is how we conclude the 9:3:1 ratio. So generally we see that the genotypic ratio is very complex, but for the NEET perspective, you need to remember, or at least you need to know how to calculate the genotypic ratio. So when we see the genotypic ratio, we will not see that how it is looking. Now these, just first, you will focus on for the phenotypic one, that how is the shape, how is the color. No, now you have to see that what is its genotype, or how is it composed. So here what I see that it is capital R, 2 capital R, and 2 capital Y. So this is the only type of the dominant one I have, so it is 1. Similarly, we have 2R and 1 capital Y, 1 small y. Now this has one smaller, so this and this will make it different again. So what is similar to this is this and this are similar. So we have only 2 in number. In all the other, there is either 2 small y or 1 small r. That cannot be the case. So this 2 is about this. So you have to calculate it, or you have to find out how many are there, and it will boil down to 1 is to 2 is to 4 is to 1 is to 2, and to 1 is to 2 1. This is a complex, but it is easy once you will start slowly start arranging ones accordingly. So what happened is that the similar explanation now is that when Mendel self-pollinated the F1 plants, he found that 3/4 of the F2 plants had yellow and 1/4 of them had green. Alright? And the yellow and the green color was segregated in the ratio 3:1. Now what he did was he took the round and the wrinkled seed shapes and also segregated them 3:1 ratio, like just like a monohybrid cross. Now coming to the law of independent assortment. What happens here is that in the dihybrid cross, the phenotypes of the round yellow wrinkled, all right, of the round yellow, wrinkled yellow, and the round green and the wrinkled green appeared in the ratio of 9:3:1. This we have seen. The ratio of 9:3:1 can be derived as a combination of series That is 3 yellow, 1 green with 3 round and 1 wrinkled. Alright, so what is basically here is that, see, the derivation can be written as it had 3 round is to 1 wrinkled and 3 yellow is to 1 green. That means it will produce 9 round and yellow. Alright, so when this and this will cross, it will produce 9 round and yellow. It will have 3 wrinkled and yellow, and it will again have 3 round and green along with 1 wrinkled green. So in other words, what we can say is that the probability, basically what we define is, here the probability of round yellow is, see, round we have 3 and yellow we have 3. So it is basically 3 upon 4 into 3 upon 4. That is, 1 is the probability of the round and this is the probability of the round. Alright, and this is of that of the yellow. So basically we are just multiplying the probability and then we can get 9 upon 16. Similarly, when we do a wrinkled and yellow. So wrinkled and yellow is 1 by 4. We got 4? That is, we have 3 round, 1 wrinkle. So 3 plus 1 is 4. Alright. So the total 3 plus 1 in both the cases is 4. That is how we got it. So the wrinkled yellow probability is 1 by 4 along with the probability. Oh, sorry, the wrinkled probability is 1 by 4 and the yellow one here is again 3 by 4. The green one will be 1 by 4. I'll just write for everything and this is again 1 by 4. Now just put these values in whatever probability it has. So again round we have, that is 3 by 4, and green we saw it is 1 by 4. Write it clearly, 1 by 4 is the probability. All right, and now what we need, green and wrinkled. Again green we have 1 by 4 and wrinkled again we have 1 by 4. So it is 1 by, what we got is 9 by 16, 3 by 16, 3 by 16, 1 by 16. So now we can just cancel all the denominators when we are taking the form of the ratio, and the result is 9 is to 3 is to 1. So this is a complete overview. P generation, or the parental generation, which had yellow round and green wrinkled. They form the gametes undergo fertilization, all right, and